So next speaker is Andres Noe. Do I pronounce correct? He's also a PhD student from the Jenner Institute, University of Oxford, and in the context of the same trial, he digs deeper and directly analyze the liver-resident memory CD8 T cell by deep immune phenotyping and transcriptomes and assess the correlates of protection. That's right?
That's correct. Great, thanks very much. So as you already heard, my name's Andres. I'm at the Jenner Institute. I'm a DPhil student. So today I'll be talking to you about some of the work that I've been doing. But first, a bit of a quick outline. I'll talk you through the background, a bit about the trial design, of which you've already heard, some of the immune phenotyping, the RNA sequencing, and the kinetics that we saw after IV vaccination. But first, it's the third day of the conference. I know we're all tired and there's some giants of the malaria field here, so what better opportunity than to do some pop quiz or trivia. So can anyone please shout out the names of the famous historical figures that died of malaria? I heard Tutankhamun. I heard that. Alexander the Great. And Nefertiti, no, the one on the right is Genghis Khan. Close, thank you though. So basically, malaria has been around for a very long time. The malarious world is shrinking, and there have been tremendous gains in the first 15 years of this century. Unfortunately, however, this progress has begun to stall, as shown here in the stall in the number of cases and in the number of deaths. So clearly we need a new tool, and obviously I'm preaching to the converted here. One such new tool is a vaccine. I'm going to be talking about the pre-recidic liver stage malaria vaccine and some of the work that we did to look for correlates of protection. So as you've already heard, I'll quickly just go through the EMI-TRAP construct. It's made up of the TRAP, so thrombospondin-related adhesion protein, and the multiple epitope string of T-cell epitopes. And we delivered these in the ChAd63 MVA heterologous prime boost. And after iterative improvement, we can see that we're able to generate a potent TRAP-specific CD8 positive T-cell response. And this is important because these cells correlate with protection, okay? Unfortunately, however, liver stage malaria, or many liver stage malaria vaccines have been unable to generate a high enough and a durable enough T-cell response to confer sterile protection. So as we've heard, this is the main impetus for Prime Target, which I won't go through again in detail, but really the main take-home message that I want you to take from this is that Prime Target can induce TRMs, and these TRMs are both necessary and sufficient for liver stage malaria protection in mice. Okay, so let's talk a bit more about my favourite type of T cell, which is TRMs, and these are tissue-resident memory T cells. So these they colonise portals of pathogen entry, such as the liver, the lung, the gut, and they sit to quickly sort of respond to reinfection. And about 6 years ago now, there was a core transcriptional signature shown in mice that's orchestrated by two related transcriptional factors. And more recently, a homologous profile has been demonstrated in humans. So really these cells exhibit increased expression of adhesion and inhibitory molecules, as well as both pro-inflammatory and regulatory cytokines, and they demonstrate a lower cell turnover than their effective memory counterparts. Than their effective memory counterparts. So again, the take-home message here is that TRMs are a specialised subset of T cells, of memory T cells, and that they have a core transcriptional signature, which they can be identified from. So again, this has been discussed, so I won't belabor it, but what I want to point out is that the Prime Target trial had 5 groups, 4 of which are shown here and 4 of which were vaccinated. All 5 received a challenge, and one of these groups had significant sterile protection. So onto the sampling for this trial. We looked throughout the trial for the correlates of protection in the blood, and then at one time point after IV viral vector, we took matched liver and blood samples using a fine needle aspirate. This was done by an interventional radiologist under ultrasound guidance, and I'm more than happy to chat to anyone that will really listen about this sampling procedure, because we were very happy about how high the yield was in terms of the cells. And then so following this, we also did some flow cytometry, and FACS sorted several populations of cells, and then we moved on to mini bulk and single cell RNA sequencing on the NextSeq 500. So what did we find? On the right, we have the markers that we use for the flow, and instead of showing you hundreds or thousands of FACS plots, I'm going to just show you some t-SNE plots to summarize what we saw. And here we have the comparison between total CD8 T cells compared to TRM memory T cells. And really, this comparison, we start to see that some clusters start to come out. And to investigate whether these clusters or some of these clusters were dependent on CD69, which, if you remember from one of my previous slides, is an identifying marker of TRMs, we compared CD69 positive to CD69 negative populations. And here we see sort of a central, I like to call it a blob scientifically, that there's plenty of overlap, and then there's no overlap down here with less overlap in these other two clusters. What about in the blood? Likewise, there's a similar chunk here in which there's plenty of overlap, no overlap down the bottom left, and then these other two clusters in which there's less overlap. So then it sort of seems that these clusters may be driven by CD69 expression, possibly amongst other things. So now if we superimpose the TRM subsets from both liver and the blood, we can see three subsets. And these three subsets sort of seem to be driven by three important TRM markers that have been demonstrated to be important in TRMs. And these three the three subsets are sort of identified by CXCR6 positive, PD1 positive, and then CD103 negative in subset 1. Subset 2 is a triple positive population. And then finally, the subset 3 is CXCR6 negative and then positive for the other two markers. Now, if we go back a step and if we sort of add these all together, then we compare the levels of these TRMs in both the liver and in the blood. We see that they correlate. So really, again, the take-home message here is that despite there being heterogeneity within the subset, there are sort of all-encompassing phenotypic correlations between the two. So to explore these differences, we looked into the RNA sequencing. So on the left, I'll show you the differences between TRMs, or tissue-resident memory T cells, and effector memory T cells, and these are from the liver. Then I'll show you on the right TRMs in the liver and in the blood. So really, this first pass plot, apart from being an absolute eyesore, and I'll apologise for that, is basically just to show you that there's over 100 differentially expressed genes. And so really that shouldn't be surprising to us, because these cell populations are incredibly different. One's resident, one's not. So what about if we punch in that transcriptional signature that I was talking about earlier? You can see here that the direction of gene expression or the fold changes concur with what's known in the liver. And so that TRM signature is important for TRM cell maintenance and survival. And if we look at the comparisons between liver TRMs and blood TRMs, we see that fewer genes involved in this signature are differentially expressed, but also if you look on the x-axis, the fold change is lower in magnitude. Another thing that I just wanted to point out here is that it's rare that we get sanity checks in bioinformatics, or it's rare that we're able to confirm sanity checks, but this here just shows us that using both liver fine needle aspirates and RNA sequencing, we're able to reliably identify TRMs from within the liver. So that's a sort of proof of concept. Okay, and so now if we want to look further into these differences, we can start to sort of categorize genes into three domains. These three domains relate to cellular location of the gene or gene product, into the biological processes involved, and then the elemental activities. What do we see? Basically, for the cellular components, we see many genes involved in the cytoskeleton, but then also structural fibers. In terms of the corresponding biological processes, we see those related to regulation of localization and movement of cellular components. And it's unsurprising, really, that actin binding is also one of the main molecular functions involved. Importantly here, we did not see differences in regulation of immune system function, in cytokine signaling, or in receptor plasma membrane receptor complexes or plasma membrane receptors. So again, the take-home message of this is that there are differences amongst the TRM populations from the liver and the blood, but those relate to mostly structural and movement-related genes. And so, again, that should be expected, because if we think about TRMs in the liver, they are patrolling the sinusoids, and they're trying to kill infected cells, which actually requires changes in cellular morphology, unlike those that circulate in the blood. So now onto the sort of flow cytometry time course. To do this, we looked at the blood on the day of IV viral vector, 1, 3, 14, and 7 days after viral vector, and then also 1 day before and 7 days after controlled human malaria infection. And so as a high-level overview, we see that these CD69 positive, CD11 alpha high memory T cells sort of expand in the blood immediately after IV, or sort of shortly after IV viral vector, and then that sort of dissipates by about day 7. And if we look at the chemokine receptor CXCR6, which is important for TRM localization to tissues, then we see that this is upregulated in the CD69 positive, so the TRM population, compared to the CD69 negative population. So again, that's another sanity check to see whether it is sort of these, in fact, that we are sampling these TRM-like cells. So then, lastly, we asked the question, if we stratify volunteers according to one of these parameters that I showed you on the previous slide, can we make predictions about protection? And in short, the answer is yes. So here we see that if we stratify half above the median and half below of day 3 TRM-like cells in the blood, then there's clear separation of the survival curves. It's a significant log rank test and 40% protected. The longer answer to that question that I asked at the start of this slide is yes and no, because unfortunately, if we look at these associations for different time points, so this is 14 days after IV viral vector, or seven days after IV viral vector, those parameters don't seem to separate out those volunteers that were sterile protected. So next we ask the question, what about if we sort of combine those parameters at an individual level so that we get more power? Eventually what we come to is a 3-parameter model in which we can significantly sort of predict the hazard of malaria diagnosis after CHMI, which is significant here. The sort of first 2 parameters are associated with a decreased risk of malaria diagnosis after CHMI, which sort of makes sense and fits in with our paradigm of them being important. Sort of puzzlingly, this last parameter is associated with an increased risk of malaria diagnosis, and so sort of perhaps this is because day 7 after challenge, so C7 means 7 days after controlled human malaria infection. That perhaps means that because at that stage it corresponds to mid to late liver stage infection, if these cells are seen in the blood, they're perhaps not where they need to be, which is within the liver. So that's perhaps why they're associated with an increased risk of malaria diagnosis. So again, we did the same sort of analyses looking at total CD8s, at CD8 positive, CD45 RA negative, so memory T cells, and then CD4 memory T cells, or CD4 cells, and there was no sort of useful models that came out of it. So again, the sort of take-home message is here that we can basically use these TRM-like cells that are circulating in the periphery to make estimations about protection. As an outline, so today I showed you that we examined correlates of protection for a vaccine candidate using RNA sequencing and flow cytometry. Liver and blood TRMs correlate phenotypically, but they differ in terms of structure and movement related genes. However, we can still manage to use these TRM levels to estimate hazards of malaria diagnosis after CHMI. I'd like to thank everyone that's involved in the studies, of which there was many, particularly Professor Adrian Hill, who's been incredibly supportive throughout this whole, and also to the funders and to you guys for listening. Thank you very much.